Over billions of years, tiny bacteria evolved a system to recognize and destroy attacking viruses. This natural defense, known as CRISPR, sparked curiosity in biochemist Jennifer Doudna and led to one of the biggest breakthroughs in modern science.
Well, I started working on the chemical basis of CRISPR protection in cells, how it works as an immune system.
Doudna and microbiologist Emmanuelle Charpentier observed a group of molecules that act like targeted scissors, snipping viral DNA in a specific spot that matches a guide sequence. Then they realized that they could perfect this mechanism for use far beyond bacteria. Swap in the right guide sequence, and you can cut and edit any genome exactly where you want.
A good way to think about it is as a surgical tool that allows rewriting the code of life.
Transformed into a technology, CRISPR has revolutionized genetic research, opening up possibilities in fields from medicine to energy, and in 2020, earning the two researchers a Nobel Prize. Doudna began her scientific journey as a child who was curious about the world around her.
I think I was in probably 5th grade in Hawaii, and growing up in the Big Island, I loved the natural world there, the plants, animals, blind spiders, etc., that I encountered in my youth. And I found myself wondering why those plants and animals had evolved to be the way they were.
She's been pursuing big questions ever since.
I love the fact that science is really about asking how do things work and then figuring it out. I still pinch myself thinking, this is my job. I think that there's a really interesting balance that we try to find between that very fundamental curiosity, just wondering, how does that work, to applying the knowledge that we get from that kind of discovery for practical solutions to real problems. So about 1.5 years ago, the Food and Drug Administration approved a CRISPR therapy for patients that have sickle cell disease. This is a devastating genetic disorder, and with CRISPR, we can now provide a one-and-done effective cure for those patients. It's amazing, and I've talked to some of the patients treated with this therapeutic. It's changed their lives in a very positive way. What's the downside? Well, a big one right now is the cost. It's $2 million a patient. I imagine a time when we can produce molecules for this therapy that are highly effective, so we need to use fewer of them. They're also deliverable by a simple injection, not a bone marrow transplant that's currently required, which is a many-week process that involves hospitalization. And once we've solved those issues, then we're going to be able to reduce the cost and also make it a technology and approach that's much more widely accessible by people.
In 2014, Doudna founded the Innovative Genomics Institute at UC Berkeley, where they're researching the evolutionary origins of CRISPR in bacteria and applying genome engineering to some of humanity's greatest problems. In a recent medical first, doctors used CRISPR to create a personalized treatment for a newborn's debilitating rare disease. CRISPR gene editing also holds promise for Huntington's disease, muscular dystrophy, cystic fibrosis, and different cancers. CRISPR's benefits could also extend to the planet.
There's a very exciting opportunity to use CRISPR in food production, thinking about how we can reprogram the genetics of plants so that they're more efficient at using nutrients to produce food crops, for example. About 1/3 of the methane that's released around the world, one of the most powerful greenhouse gases, comes from animal farming. Wouldn't it be great if we could effectively just reprogram the bugs that are responsible for making that methane so they don't do that?
With real-world impact comes real-world responsibilities. That's especially true when talking about changes to the human germline, sperm, eggs, and embryos that could be passed on to future generations.
That's a profound use of a technology like CRISPR, and there's no question that it requires very careful thought and analysis about when and for what purpose would such a use be warranted. Over the years, I've really advocated transparency around the use of CRISPR in the germline. I think that there may come a time when it's safe enough, we're not there today, but safe enough to use it in that setting that there may be real applications that would be ethical in the sense that we're preventing terrible disease from being transmitted. But boy, it's certainly going to require careful analysis about that to make sure it's done responsibly.
For her part in developing a technology with untold power, Jennifer Doudna is a winner of our 2025 Genius Award.